I would like to welcome Nicole and you all to the live Q&A session of the seminar. Please ask your question in the box on the screen and we will ask the most popular questions. So if there is one you would like to see answered, please use the voting function. So thank you so much for your talk, Nicole. And let's start with the first question. So you mentioned that your studies use a large cohort. What is the ideal size of cohort to extract this information? Do we still need to go larger and larger?
Thank you, Rosa, for the great question. The short answer that it really depends on what you want to do. There are obviously different size requirements for different purposes. So what we learned in complex diseases is that if you want to study, complex phenotypes, and particularly if you then want to also study effects such as for those of the environment, large population sizes are needed. So UK Biobank is obviously the reference resource for not only empowering discovery complex trait associations, but also begin to disentangle this effect of genetics and environments. For other types of phenotypes, we have also learned that much smaller sample sizes are equally valuable. So for molecular phenotypes that are closer to the effect of the gene, for instance, gene expression phenotypes or indeed metabolites, you can get similar strength of associations in sample sizes of, a few hundred to a few thousand individuals. So really depending of, and there seems to be a rough correlation between how close the need of a sample size versus how close the phenotype is to the primary effect of the genetic variant and how complex the architecture of that particular phenotype is. So if you were to design, for instance, a functional genomic study for instance, looking at gene expression phenotypes in primary cells, you would want to perhaps maintain the sample sizes as small and use if you want your capital to, for instance, go to multiple populations. And obviously the aim ultimately is also for large scale cohort studies to capture the whole diversity of human genetic variation. So not only European populations, but indeed in all the major population groups in the world. So with respect to this, it's been great that new resources are coming online, including last week a GWAS of the Taiwanese population with hundreds of thousands of people, and similar size cohorts are being developed in other parts of the world.
Thank you.
Thanks, Nicole. It's a great talk. You talked a little bit about polygenic risk scores, and I just wondered, what's the thinking about looking at interaction terms between those different features and those polygenic scores? And in general, I think I certainly have an interest in genetic interactions and Mendelian diseases, and what is the evidence for them, and maybe what are some of the barriers to detecting them? Yeah,
so the use of polygenic risk scores for complex trait disease predisposition so far are mostly accounting for additive models of inheritance, which is effectively what we have discovered so far in association studies. Still very, very much difficult, even with large sample sizes, to detect interactions if they exist. Then the next question is whether there are interactions across different types or layers of genetic data. So, for instance, does the variation in germline genome interact with somatic mutations, or whether there's non-additive effects between Mendelian mutations and polygenic risk scores? And I think, as far as I know, there's really not a lot of evidence yet, but this is beginning. The example like the rare disease examples, that I've shown are the first times where we're beginning to be able to explore and detect a significant effect on polygenic. Polygenic risk scores are becoming sufficiently powered that we can begin to detect effects, for instance, against a rare disease. And so this is also the first step to then look for deviation from additivity. So, the short answer is that we don't know yet, but I think it will be really exciting to test in the near future.
And is it, because it's very interesting that historically that they haven't been detected, certainly in complex diseases. Is it your intuition that it's an absence of power, or do you think fundamentally they're not going to be that important? I'm coming from an oncology background where we clearly know the presence of different somatic variations has a very important impact on disease, in many aspects of disease.
Yeah, again, it's difficult to be conclusive about this, but what we can say is that given the power that we had so far, we haven't been able to see. So if these interactions exist, they will be weaker than what we can currently see with current data sizes.
Okay, thank you.
Thanks, Nicole. We've got a question that's come in from the audience. What do you see as the best strategy to validate associations with non-coding variants? And which are the vast majority of these? Where are these associated with different diseases? So what's the best strategy to validate associations with non-coding variants?
I guess for validation in this case is meant to really understand the function rather than just as a technical validation. So in the case, obviously, what really many groups in the world are doing is really to use this functional genomics toolbox as a very generic term, that can mean many things, to really begin to first of all collect observations that allow us to annotate these variants more efficiently and as accurately as possible. And that information can come from many different sources, from maps of regulatory QTLs. They can come from assays such as multiparallel reporter assays, CRISPR-based assays, readouts of integration with epigenome maps, etc. Then the next stage is to switch to perturbation type experiment like CRISPR, where we actually engineer these genetic variants and we look at downstream effects, on gene expression or on other cellular phenotypes. That's the next step in terms of how laborious this is, but it's a system where we can control that you're really looking at a direct effect rather than simply a random chance overlap. And these efforts are becoming scalable. These techniques are becoming scalable, and they are providing us with in the near future for sure the ability to go into the testing of hundreds of variants, if not thousands at once. And then the next complexity to that is also understanding which cell types these variants are acting. So being able to do these experiments in the right cell type. So historically, we relied a lot on model cell lines and potentially now induced pluripotent stem cells. But really, ideally, what we want to do is experiments that are in primary cells, and ideally assaying the whole range of primary cells that are potentially responsible for that phenotype. So these are all screening. And indeed, one of the the goals of the ICDA, the International Common Disease Alliance, is really that to create a framework by which we can do these experiments in a scalable way. Obviously, the next step then is really to take each variant and really demonstrate the function. And these are multi-year experiments requiring complex experimentation.
Brilliant, thank you.
Yeah, so following in that question about validation, so yeah, these genetic studies are identifying variants and genes associated with disease risk, but how do you know whether that they may be a good point for therapeutic intervention? Do you need like more functional assays or?
Yeah, so this is, again, a hugely important point. So industry is very much focusing on these questions, together with the, academic community. And obviously, the first requisite is that that particular variant, so what gene, is the effector gene for that particular variant. And that's not always straightforward, as we know in GWAS. And so many times these functional annotation experiments are really pointed to that. Then you want to know that the gene controls a target that is causal to the phenotype. So effectively that, if you were to modify that target, you would modify the risk of disease. And that's obviously, again, difficult work to do that we're beginning to do with the Mendelian randomization approaches, where we effectively test that, for instance, the risk factor is on the causal path to disease. And then the other thing you want to do is that you also to know that if you modulate that target, you won't have side effects that are worse than the disease itself. So again, now there are in silico approaches that are looking not only modulation as a potential viable therapeutic option, but also at the fact that it doesn't provide, in silico provide adverse effects. So there are wonderful work that has been shown by many colleagues, and one that springs to mind, there's Robert Plenge, who has really done a lot of thinking about how to do this. So the idea, for instance, is that if you identify a allelic series where you have multiple genetic variants and independently target the activity of a gene, you can use those to model in silico even the likelihood that modulation of the target will be effective.
Great, thanks, Nicole. So you mentioned UK Biobank and the interval study in your presentation, and these are fabulously rich resources. I guess just the question is, are there similar efforts in other countries, and is the long-term vision, if there are, to bring these all together, and how does one do that? What are the kind of challenges you would face to integrate biobanks across different countries?
Yeah, this is a really important and topical question. Yeah, there are large-scale resources like I mentioned Taiwan recently. China has a similarly large cohort. Japan Biobank is equally rich, and then Korea has large-scale genomic resources. And so the challenge, already we're learning a lot about combining data from these resources, and obviously we have a better understanding what predisposing alleles are shared across populations and what are not. Certainly we know that common alleles tend to on average be more shared than rare alleles, so the picture will change once we apply sequencing at scale. What is really important is though that we really now work together to tackle diversity in more underrepresented populations and ethnic groups, and so obviously African ancestry populations are partially captured to African ancestry cohorts in the US, but there are also big efforts in Africa themselves to the H3A network to collect this kind of data. Data in South America, so there are efforts now to begin to put together resources, for instance, for South America. So in fact, one of the missions of ICDA, which is indeed that to, if you want, level up the representation in genomic data sets across the world and to facilitate that progress. So ultimately, once we, we will not only learn, so doing that and achieving this representation will not only tell us a lot more than we know already about genetic risk, and particularly, for instance, the ability to find map causal variants, but also is really the only way to then really realize an equitable benefit in medicine through genetics. Because we know very well that at the moment, even clinical trials are not designed, in an equally balanced way.
Yeah. But so yeah, it's clear that you need these multiple biobanks, and they will help, for example, identify the rare alleles will change and so forth, the rare disease-associated alleles. But how are you going to do that? How are you going to bring all this together? Because actually, the power really comes if you can actually integrate them as a single mega cohort, so to speak, as opposed to a series of smaller ones. So are there ethical issues, legal issues, logistical issues? I mean, can you touch maybe just briefly on one or two of those?
Yeah, I mean, this is yes, there are already efforts to bring this, and the person that springs to mind that is spearheading some of these efforts is Mark Daly, with through the leadership in multi population cohorts, but he's by far not the only one. I think there are multiple, we learned it in the GWAS here to work with the interoperable data sets. So working not so much on on raw data, but indeed on summary statistics, which is an easier, perhaps more acceptable way to share data across national boundaries. Then and it is possible, it is likely that that will continue to be the model moving forward. Then obviously the next challenge is that to make sure that the data is harmonized in a way that makes it, interoperable, compatible. So, this setting standard for genomic data sets and for genetic analysis is hugely important for achieving that. So, having broad consensus that allows as much as possible model of data sharing, like UK Biobank, is desirable. It will not be applicable everywhere. But then the next best thing is really this ability to create standardized data sets that can be then merged. And obviously the standardization is not only on the genetic side, but indeed also on the phenotypic side. So the use of, for instance, standard clinical, disease codes or phenotype definitions.
Sounds like a very big job. Thank you.
I'm going to ask, where do you believe efforts should be prioritized in the future? Should it be larger disease cohorts for GWAS, or richer phenotyping of samples, or functional assays? Or are you going to say all of those?
Yes, I mean, again, depends very much on where the starting point is. So I think it is very clear priorities to extend the genomic data sets to non-European populations, and we said that before. We are learning, as we do analysis, we learn a lot about what is really the useful sample size for a functional genomics, for instance, data set. And my expectation is that in the area, efforts in the future will be directed especially to try to cover as much of, for instance, the cellular or the tissue diversity of the human body, and at the same time, continue to embrace new technologies as they become scalable and approachable to as many data sets as possible. So that will be the priority. And it is very clear now that these experiments need to be done whenever possible, again, in a multi-population setting. The phenotyping, it is hugely challenging to obtain deep phenotypic data sets such as UK Biobank in interval is extremely costly. So it is very unlikely that we'll be able to achieve the same depth of phenotype in every population. But obviously the ability to, for instance, develop systems that make electronic health records interoperable with, will be a good way to achieve at least some basic level of phenotyping in large sample sizes. So I think we probably move towards a model by which there will be a few choice resources that will be very deeply phenotyped, but we hope to achieve extensive phenotyping through, for instance, data from medical records already collected.
Thank you.
Yes, and thank you so much. I wonder if you can tell us a bit of how much the structural variants added to your PRS study and how significant was their effect. And in general, do you think it's something that we should be doing for genetic studies?
Yeah, that's also a very, very good question. We haven't really incorporated structural variants in our PRSs at all, so I don't really have a direct answer. So we know that many common, so most of the PRSs so far really only capture common variation, and we know that many structural variants are in LD with the SNPs. So it might end up that incorporating structural variants for common variants might not add too much to PRSs themselves. It might add to the interpretation of the causal effects. Obviously, as we move to characterize variation in not so much the rare, but perhaps more the low frequency end of the spectrum, then there might be gains there. But we haven't done an experiment, and I'm not sure anybody has done it.
Great, and I think we have time for one more short question. I think you made a point about the fact that you often see individuals who are homozygous for recessive disorders yet apparently healthy. Do you think it'd be worthwhile kind of following those up or pursuing those further as interesting case studies?
Yeah, I think so. I mean, UK Biobank doesn't have an option to recontact individuals, so it would be difficult to so this is a limitation, a characteristic of biobank. For sure, one of the limitations of our studies that is relying on variants that are imputed. So, we can impute some of the classical clean variants, but by no means we have a complete picture of variability. So I think the first experiment there will be to take whole genome sequencing as data as soon as it's generated and look for additional variants. And so that would allow us, for instance, to tease apart the cases where there might be another clinical variant that has been not imputed. So, this is, so if you wanted, this is a really best case scenario, but I'm sure that the genome sequencing will provide us with a much greater resolution in that sense. I think then the question is, so do we believe that PRS alone can generate rare disease? And so I think this is the next question. There's clearly an association, a positive association, or potentially a protective effect or a risk effect, but is likely to be more of in the realm of modifier rather than driving susceptibility alone. But it's something that we absolutely want to explore further.
Great.
I think
Thank you.
We certainly can think this is could be, for instance, a good application to at least reassess the diagnosis on the basis of the underlying polygenic risk.
Great, thank you, Nicole. I'm afraid we are out of time and the hour is now up. But I'd just like to take a moment to thank you so much for preparing your presentation and taking the time to meet and discuss this with us today. And just to tell the audience that we're going to continue these seminars through the next few months and to the end of the year. Our next seminar will be May the 19th. There will be an announcement shortly about who that speaker will be. And really these seminars themselves are archived on the Sanger web pages for you to view at your leisure and to revisit whenever you'd like. And we're just really pleased that we could have this opportunity to share Sanger Science with you. So thank you very much for joining, and we hope to see you next month. Thank you.
Thank you. Thank you, everybody.